In galactose metabolism, galactose is converted into glucose so that it can be used in glycolysis from the beginning of the pathway. This mostly takes place in the liver, but other tissues also metabolize galactose to a lesser extent. Normally, dietary lactose is broken down into galactose and glucose. Next, galactose is phosphorylated by the enzyme galactokinase to produce galactose 1-phosphate, which is converted by uridyl transferase to glucose 1-phosphate. This can be converted to glucose 6-phosphate, which can be used to start glycolysis or finish gluconeogenesis, depending on the energy needs of the cell at the time. What happens if you're missing some of the galactose metabolizing enzymes? Galactokinase deficiency is an autosomal recessive deficiency of galactokinase, and instead causes it to be converted to an alcohol form, galactitol. This is catalyzed by the enzyme aldose reductase, the same enzyme that also converts glucose to sorbitol, which we'll see in the next slide. Please don't confuse this with aldolase B. Galactokinase deficiency is usually a mild condition, but galactitol does accumulate inside of cells, and can cause galactosemia, infantile cataracts, and failure to track objects or develop a social smile. Think of it this way. If an infant can't see a face, then it can't elicit the reflex of the social smile. Well, wait a minute. How does this cause galactosemia if we just said that galactitol accumulates inside of cells? Well, think of this as clogging up a sink. If there's nowhere for the ingested galactose to go, it will accumulate and spill out into the serum. Now, classic galactosemia is a more serious autosomal recessive deficiency of galactose 1-phosphate uridyl transferase. A deficiency of galactose 1-phosphate uridyl transferase causes galactose 1-phosphate and galactitol to accumulate inside of cells, causing some of the same effects as galactokinase deficiency, such as infantile cataracts, in addition to some new signs due to the galactose 1-phosphate accumulation, such as jaundice and hepatomegaly. These are similar to what you see in fructose intolerance, since in both cases you're accumulating a phosphorylated monosaccharide inside of hepatocytes, resulting in liver damage, while at the same time depleting intracellular phosphate stores. The treatment for classic galactosemia is simple. Don't eat anything with either galactose or lactose, which is a disaccharide made up of galactose and glucose. Sorbitol, which is shown here, is a reduced alcohol form of glucose. Sorbitol is formed by the enzyme aldose reductase. This enzyme should sound familiar since this also converts galactose to galactitol, as I mentioned earlier. So sorbitol is created by aldose reductase. When glucose gets converted into its sorbitol form, it becomes trapped inside of cells, and can later be converted to fructose via sorbitol dehydrogenase. Now, not all cells in the body have the enzyme sorbitol dehydrogenase. Fortunately, the liver, ovaries, and seminal vesicles contain both enzymes aldose reductase and sorbitol dehydrogenase. However, in tissues that lack sorbitol dehydrogenase, to break down the sorbitol, toxic levels of sorbitol can accumulate inside these cells, which causes damage because of sorbitol's osmotic activity. Now, what do we mean when we say that something is osmotically active? Well, in this case, high amounts of sorbitol can create a concentration gradient, pulling water into the cell, causing it to swell. In states of persistent hyperglycemia, such as in diabetes, high sorbitol levels can cause damage to the lens, the retina, kidneys, and Schwann cells, since these all only have aldose reductase and not sorbitol dehydrogenase. This can cause cataracts, retinopathy, glomerular disease, and peripheral neuropathy, all of which are unfortunately common issues in diabetes. Okay, flash quiz time. A poorly controlled type 2 diabetic man has poor vision and tingling in his feet. How can sorbitol be at fault here? Well, this is because chronic hyperglycemia or high blood sugar levels causes sorbitol to accumulate and cause osmotic damage in these cells. This is most prominent in the cells that lack sorbitol dehydrogenase to break down the sorbitol, including Schwann cells, the lens, the retina, and the kidney. Have you ever wondered what someone meant when someone says they are lactose intolerant? Well, we'll get to that in a sec. Lactase is a brush border enzyme on the luminal side of the small intestine, which hydrolyzes or splits the disaccharide lactose into its constituents, glucose and galactose, which can then be transported into the enterocyte. A deficiency in this enzyme can either be primary or secondary. A primary deficiency is due to aging and losing expression of the lactase allele, usually seen in people of Asian, African, or Native American descent. Lactase deficiency can also be secondary, which is usually transient following a bout of gastroenteritis or from an autoimmune disease. Lactase deficiency can also be congenital, but this is usually rare. How does a lactase deficiency typically present? Symptoms of lactase deficiency and subsequent lactose intolerance include bloating, cramps, and osmotic diarrhea. You'll see these symptoms because lactose won't be absorbed into the enterocytes and therefore will pull water into the lumen of the GI tract due to the osmotic gradient. Bacteria in the gut can then use this lactose to form methane, leading to flatulence. You can diagnose lactase deficiency clinically, but stool testing will demonstrate a decreased pH and breath tests will show increased hydrogen content. Let's say you're attending orders an intestinal biopsy to determine the cause of the patient's severe persistent diarrhea and cramps. What would these biopsy results show in this case? Well, hopefully you're thinking that the biopsy results will be normal. Otherwise, you're wishing harm onto your patient, and that's kind of rude. Okay, remember that lactase is a brush border enzyme. If it's deficient, there'll be a lack of enzymatic activity at the brush border, but the GI epithelium will still look normal under a microscope. But this is assuming that this is a primary deficit. If the intolerance is secondary, there may well be damage. However, in the case of primary lactose intolerance, again, the biopsy will appear normal. Treatment for lactase deficiency includes avoiding lactose in the diet or taking lactase pills with food. So far, we've covered the concepts of carbohydrate metabolism. Now we'll move into proteins and their building blocks, amino acids. Only the L form for levo, meaning left, of amino acids are found in proteins. As you might remember from undergraduate biochemistry, amino acids can be categorized by whether they're essential or non-essential, and whether they're acidic, basic, or neutral. Essential amino acids have to be supplied in the diet, since humans can't produce them. The glucogenic essential amino acids, which is a fancy way of saying that they can be converted to glucose via gluconeogenesis in the liver, are methionine, valine, and histidine. Repeat that a bunch. Copy it down, do whatever you gotta do to remember it. The essential amino acids that can be either glucogenic or ketogenic include isoleucine, phenylalanine, threonine, and tryptophan. Do you remember what the purely ketogenic amino acids are? The purely ketogenic essential amino acids, which can be converted to ketone bodies, are leucine and lysine. Again, the purely ketogenic essential amino acids, which can be converted to ketone bodies, are leucine and lysine. Do you remember what enzyme deficiency requires purely ketogenic amino acids as a treatment? Well, hopefully you remember pyruvate dehydrogenase deficiency, which requires giving the ketogenic amino acids to bypass the deficient or defective enzyme pyruvate dehydrogenase. Also, some amino acids are considered to be partially essential, or only essential sometimes, because we can synthesize them in our bodies, but sometimes we can't synthesize enough to meet metabolic demands. So we'll need a little extra from our diet. In particular, arginine is usually not required in adults, but kids can't produce the amino acid arginine fast enough to keep up with their growth. So in this case, arginine would be required in their diet. Just remember that all other amino acids that I don't cover are the non-essential amino acids. We also have acidic amino acids, kind of redundant, but roll with me here, which have a negative charge at a physiologic pH. Remember, that means between 7.2 and 7.6. There are only two acidic amino acids, which are aspartate and glutamate. The alternate name for these two amino acids are aspartic acid and glutamic acid. Well, go figure. The basic amino acids, which are usually positively charged at physiologic pH, are arginine, lysine, and histidine. Arginine is the most basic, and histidine does not actually have a positive charge at physiologic pH. It's kind of a weirdo. Again, what's physiologic pH? Remember, it's about 7.4. We can incorporate our discussion earlier about histones, the molecules that DNA wraps around in the nucleus. Remember that DNA is negatively charged, so the histones can increase their composition of positively charged or basic amino acids to attract and hold on to the DNA. Interestingly, the only basic amino acid that sounds like histone, which is histidine, is the only basic amino acid that's not really used for this purpose. Alright, so we just talked about the different kinds of amino acids. Now, let's go through how they're broken down. As we've seen, not all amino acids are created equal. Hashtag overused cliche. Different amino acids go through different catabolic processes, many of which yield metabolic fuel in the form of pyruvate or acetyl-CoA, and ultimately glucose and ketone bodies, but these pathways are not very high yield for the exam. If you're on your second or third pass of the material and you feel like you have time, maybe look at these. Otherwise, keep going. The degradation pathways of all amino acids all produce ammonia, though, and if this isn't disposed of properly, it can cause problems. So how does the body get rid of ammonia, pictured here as the ammonium ion? Urea is the relatively non-toxic, disposable form of ammonia, which is synthesized in the liver and excreted via the kidneys into the urine. The urea accounts for about 90% of all nitrogenous compounds excreted in the urine. The reactants for the urea cycle are ammonium, CO2, shown here as bicarbonate, and ATP. And the products are urea, ADP, and fumarate. It's important to note that the urea cycle takes place in both mitochondria and the cytoplasm. The rate-limiting step is the first reaction, which is catalyzed by carbamoyl phosphate synthetase. This enzyme converts carbon dioxide and ammonium ion to carbamoyl phosphate, which can then enter the urea cycle and combine with ornithine to create citrulline. The enzyme carbamoyl phosphate synthetase may sound familiar since you heard it at the beginning of the biochemistry chapter. Just remember that carbamoyl phosphate synthetase 1 is used in the urea cycle, where carbamoyl phosphate synthetase 2 creates carbamoyl phosphate in pyrimidine synthesis. Here, once we create a molecule of carbamoyl phosphate, we're ready to take a spin in the urea cycle. Carbamoyl phosphate combines with ornithine to create a molecule of citrulline. We sprinkle in an aspartate and an ATP to create argininosuccinate and ADP. Before we keep cycling through, let's talk briefly about the structure of urea. Here's a mnemonic to help you remember the order of the byproducts in the urea acid cycle. Before we keep cycling through though, let's talk briefly about the structure of urea. Urea itself has two nitrogenous groups. One of the nitrogen groups comes from the ammonia that we started with, and another nitrogen group comes from the aspartate that we just added. The carbon in urea originates from the CO2 we originally added. Eventually, we get our first product out, a fumarate. Where have we seen fumarate before? Well, hopefully you're thinking about the TCA cycle, shown here for reference. Notice how fumarate can feed back into the TCA cycle so it doesn't get wasted, as opposed to me later this weekend. But back to the urea cycle. We notice how our arginine gets turned into an ornithine, which is used to restart our urea cycle. We additionally get a urea molecule out, which will travel to the kidney for excretion. Notice how it's now water-soluble and neutral. Rest easy. We've successfully rid the body of ammonia by converting it into something that we can actually filter. Woo! Okay, now we can rest easy, because we've successfully rid the body of ammonia by converting it into something that we can actually filter. Okay. If you're following along in the First Aid Step 1 book, take a look at the urea cycle figure. It's important to note that this process requires 2 ATP. I'll be discussing urea cycle disorders in just a second, so bear with me. Let's take this one step at a time and first test what we just learned about the urea cycle. Okay, flash quiz time. The urea cycle ultimately yields blank, which is excreted in the urine, and blank, which can enter the citric acid cycle. I'll give you a second. The urea cycle is meant to excrete urea into the urine, which gets rid of nitrogen in the form of ammonia formed in the body from amino acid breakdown. It also creates fumarate, which can feed into the citric acid cycle. Now that we've figured out how to get rid of ammonia, let's quickly find out how we get the ammonia produced from the body to the liver cytoplasm. As I mentioned before, ammonia is toxic and must be converted to urea so that it can be excreted in the urine. However, the urea cycle only occurs in the liver, which means that ammonia that gets created by the periphery must be transported to the liver first to be able to get rid of it from the body. Now, how does the toxic ammonia get from the body to the liver? This transport occurs in the form of alanine, which is shown here. In the muscle, the amine group that you're trying to dispose of is added to alpha-ketoglutarate in a transamination reaction, which results in glutamate. By the way, this is the same alpha-ketoglutarate that we talked about in the TCA cycle. In a second transamination reaction, this amine group is transferred to pyruvate, forming alanine. This is catalyzed by alanine aminotransferase, or ALT. one of the same enzymes that you measure for hepatic function testing. Alanine can then circulate throughout the body through the Cahill cycle. Once in the liver, the previous reaction is reversed, and the amino group is transferred from alanine to another alpha-ketoglutarate to form glutamate. In the final step, the ammonium ion from glutamate enters the urea cycle and is used to make urea. This also allows glutamate to cycle back to form alpha-ketoglutarate.